Hello everyone, welcome back to Guideline Gurukul, your go-to place for breaking down complex pharmaceutical guidelines into simple, easy to understand concepts. In today's session, we'll be discussing F1 and F2 in dissolution studies, an important area that every pharma student and professional should know. At the end of the video, I will give you an interview question relevant to this topic. So, let's get started. Before going to this topic, I want to make it clear that it's about F1 and F2, not F1 and F2. Yes, there is a difference between lowercase and uppercase F1, F2. So, what's the difference? F1 and F2 are similarity factors used in dissolution profile comparison between a test product, usually a generic, and a reference product, that is R&D. Whereas F1 and F2 refer to phase 1 and phase 2 clinical trials in drug development, that is clinical trial phases. That's a separate video I need to make. Before you understand about F1 and F2, we need to discuss about dissolution. When you swallow a tablet, it needs to break down and dissolve in stomach or intestinal fluids. Dissolution test measures percentage of drug dissolved at different time intervals like 10 minutes, 20 minutes and 30 minutes. Regulators like FDA, EMA, etc. wants us to compare the dissolution profile of products whenever required. But why to compare it when it is required? Well, there are generally two cases to consider when we compare the dissolution profile of drugs. First case is to show that a generic drug is equivalent to brand drug. When a generic drug is developed, we need to prove that it is equivalent to brand drug. In such cases, we need to compare the dissolution profile of generic drug with the brand drug. Second case is to show that a product after manufacturing changes behaves same as before. See, once the product is approved and whenever we want to go for further changes of the approved product, we need to show that the product after changes we did behaves the same as before. The dissolution profile in these two cases can be compared based on two factors, that is F1 and F2. Let's learn about it. F1 is called the difference factors. It shows how different the two dissolution curves are, that is the generic drug and the brand drug curves. The smaller the number is, the similarity is more between the curves. If the number is larger, the difference is more between the curves. F2 is a similarity factor. It gives a score from 0 to 100, showing how similar two curves are, that is, two dissolution curves are. The bigger the number is, the more is the similarity. The smaller the number is, the less is the similarity between the dissolution curves. Let's learn about magic numbers. To know if the drug product dissolution profiles are similar or different from each other, we need to understand these magic numbers. As per ICH, FDA and EU guidelines, if F1 is less than 15 and F2 is more than 50, the two products are considered to be dissolved in a similar way. However, there is another condition here. If both products dissolve more than 85% in 15 minutes, then their dissolution are considered similar and we need not do F1 and F2 for this. These are the formulas to calculate F1 and F2. RT is percentage dissolved of the reference drug at time t. TT is percentage dissolved of the test drug at time t, where n is number of time points excluding 0. Want to remember the formulas? Well guys, please don't stress about memorizing the formulas. Just remember that F1 is difference and F2 is similarity score. Now, as promised, we will discuss an important interview question with respect to F1 and F2. Just why are acceptable values of F1 and F2 15 and 50? Why not have some other values? And for answer this is, if F1 is 15, it means the average test curve differs by 15% of the reference values. Regulators like FDA, EMA or ICH decided that a difference of less than 15 is non-clinically meaningful. In other words, if your tablet releases drugs within 15% of the reference at each point, the absorption in humans will likely be the same because in vivo variability is much larger anyway. In short, for F1, a difference of 15% or less is considered small or not clinically relevant. Similarly, for F2, if the average difference between two profiles at each point of time is about 10%, then F2 will be 50. Above 50 is the difference is less than 10%. Exactly 50 is 10% like an average. Below 50 is more than 10% difference. For F2, a score of 50 corresponds to about 10% average difference, which means acceptable, because it usually means the product behaves in the same way as in vivo. These are the values selected by regulators after testing on many datasets and balancing clinical relevance with practical acceptance. That's all for today, guys. I hope you found this video useful. Suggest me any other interview questions you have faced and we can discuss. We can have a very good discussion in this chat forum. We will meet again with another important topic. Please like and subscribe to my channel. This is Meg. Thank you so much for watching.